Alright, hello everyone. I'm going to be talking to you today about the reverse problem from the one we've been talking about for several lectures. So we've been talking a lot about tearing apart carbonaceous molecules to generate energy, and one of the goals of the cell is to do something with that energy, and one of those things is to make cellular components to essentially grow and divide. And that process is biosynthesis. And this is a fundamental set of biochemical reactions that are involved in anabolism, or essentially the reverse process of, instead of oxidizing carbon, that we're going to reduce carbonaceous compounds and sources of nitrogen, etc., to produce useful products for the cell to build new cells. And so, again, here's this big picture that we've been going through for several lectures, right? And that essentially we have glycolysis, that we have generated a lot of, liberated a lot of electrons from oxidation of things like glucose, and we have demonstrated that we can fully oxidize those in the citric acid cycle, or we can use fermentation as an option if there is no oxygen around. And electron transport is one of the places that there's a great diversity in what microorganisms do. And so what I want to illustrate today is to fill in a lot of these other arrows. So we've focused a lot on this sort of central set of reactions to take glucose down to CO2, how we generate energy, and ultimately there's a lot of other things going on, right? So some of the things that we want to be able to do are to look at basically how does a cell eat amino acids? How does a cell make amino acids? How does a cell use nucleic acids? How does it make nucleic acids and lipids? So what I want to do today is to make sure that we have all of the pieces we need. And to do that, we're first going to need to address a few pieces of catabolic biochemistry that we did not cover yet. Specifically, how does a cell eat lipids, and how does a bacterial cell, eat amino acids? And so these are things that are routine parts of a microorganism's diet. So ultimately, the big question for today is, given defined sources of carbon, oxygen, nitrogen, phosphorus, and sulfur, and all the other trace things that we talked about, the nutritional composition of what makes a cell, how does a microbe, we'll be focusing on prokaryotes here today, make all of the macromolecules it needs to make a new cell? So the first thing we need to talk about is how are lipids consumed. And this is through a very important set of reactions called beta oxidation. And so what's essentially going to happen is that the lipid that is fatty acids bonded to glycerol through ester linkages, those ester bonds will be cleaved by an enzyme called lipase. That glycerol is then catabolized by stage 2 of glycolysis. It's essentially a 3-carbon compound. And that the fatty acids then require a different pathway that results in production of a great deal of acetyl-CoA. So, breaking this down into what I just said, essentially these triacylglycerols, for example, will just take a simple fat, a simple lipid, and lipases are hydrolase enzymes. They hydrolyze off fatty acids and from glycerol. So glycerol is something that can immediately be put into glycolysis through a single preparatory reaction, and it actually makes dihydroxyacetone phosphate, which is one of the 3-carbon precursors that essentially begins stage 2 of glycolysis. Now, fatty acids are a different beast. They're different molecule and require different biochemical reactions to break this down. And there's a very special pathway that is conserved across just basically about any organism, and that you need to be able to make lipids and you need to be able to break them down, because lipids are actually an extraordinarily rich source of electrons. So all of the carbons in that fatty acid tail are fully reduced carbons. So if you have a way to take those electrons off, it's a great great way to generate a lot of reducing power in the cell that can be used to generate ATP, or it can be used to generate biomass. So what happens in this process of beta oxidation is that when we look at a fatty acid, there's the carboxyl group, and next to this carboxylate group is what we call the alpha carbon. And next to that, in a very non-creative way, is the beta carbon. And so the process of beta oxidation is essentially oxidizing this second carbon sequentially so that essentially this carbon-carbon bond here can be more or less broken through this carbon-carbon bond here can basically be broken by the introduction of acetyl-CoA. So like many of the biochemical reactions that we've seen, especially glycolysis, there often is a preparatory reaction, that there has to be some initial energy investment to activate these substrates for subsequent oxidation. And this is no exception. So beta oxidation, what it does is this initial reaction, this CoA activation reaction, uses free coenzyme A and input of ATP to ligate CoA onto this fatty acid. Now this fatty acid is now a fatty acid CoA, and now we can begin the process of beta oxidation. So this is an activated molecule. It's got a big molecular handle and essentially is allowing electrons to more freely move around these carbon-carbon bonds. And with this happening, there is now an oxidoreductase that is FAD dependent, and in the process of transferring electrons to FAD to produce FADH2, you now oxidize this carbon-carbon bond between the beta and alpha carbon, and now water is added across this bond, this double bond. So essentially now this beta carbon is in its first oxidation state, carrying this hydroxyl group. A second oxidoreductase enzyme oxidizes this same carbon one step further to put this carbon into its second oxidation state to form a ketone. So now we have essentially two oxidized carbons that now another essentially ligation type reaction can happen where CoA is used as a substrate to essentially break this carbon-carbon bond to liberate acetyl-CoA and to produce a carbon chain that's almost identical to what we saw up here, except it's 2 carbons shorter. Now acetyl-CoA, as we saw with the citric acid cycle, this acetyl-CoA can be oxidized to CO2 to liberate 8 electrons, essentially. So this beta oxidation process, again, where it's getting its name, is that it's oxidizing this beta carbon through a sequential set of oxidoreductase reactions that are producing FADH and NADH that can be either used in the in an electron transport chain or used as reducing power to build cellular biomass that we're going to talk about in the coming slides. So if we break down what's going on with beta oxidation, I think it's important to get some perspective on how much energy is actually being liberated through the oxidation of fatty acids, and specifically lipids. So let's do some molecular accounting. So oftentimes cells will have 16 carbon tails on their fatty acid chains. And this 16 carbon fatty acid is known as palmitic acid. And so we'll imagine we have a palmitic acid phospholipid. And so for each one of these tails, there are 16 carbons, and what will happen are 8 rounds of beta oxidation. And as a result, we'll produce 8 NADH, 8 FADH2 per tail. And so there are 2 tails in a normal phospholipid. So that means that we're going to get 16 NADH and 16 FADH2 per lipid. And so, contrast that to what's going on with glycolysis, and you see that there's a lot of electrons that are liberated and can ultimately be used to generate a lot of energy. So this is one of the reasons that fats are actually so energy rich, and one of the reasons that we like butter on toast. So what you can now do, if we're going to talk about really extracting as much energy as possible, there are 8 acetyl-CoAs that enter the citric acid cycle, and there were 2 tails that we essentially had all of those acetyl-CoAs from. So from one single tail, you'll get 8 ATP, 24 NADH, and 8 FADH2 per tail of this phospholipid that we're talking about. So if we consider both tails, you just multiply all of this by 2. So now, if we think about the glycerol that was released, so glycerol phosphate, if we're going to oxidize that via the rest of glycolysis and the citric acid cycle, if we do our accounting, we'll get 7 NADH, 1 FADH2, and 3 ATP from essentially burning this glycerol. So the net energy yield from the entirety of oxidizing one whole entire phospholipid is 71 NADH, 33 FADH2, and 19 ATP. And that is a massive, massive amount of energy. So if we think about just net ATP, so imagine all of these cofactors were converted into ATP via electron transport and oxidative phosphorylation. With oxygen as a terminal electron acceptor, that ends up to be around 300 to 330 ATP per lipid. And so that is an absolutely massive amount of energy from one single lipid. And membranes are filled with these lipids. So when we go back and think about energy storage compounds, this is one of the reasons that organisms store fats. And so, why arguably we love eating fats in our own diet because they're so energy rich. So, I talked to you about how to eat essentially lipids and fats. Another major source of a microbe's diet are often amino acids. And so many amino acids are aminated intermediates from the citric acid cycle, glycolysis, and the pentose phosphate pathway. So what that means is that if you're able to remove the amines from amino acids, the carbon skeletons that are left behind are often readily metabolizable by those big major pathways that I showed you in that diagram above. So amino acids used as catabolic substrates are produced by microorganisms hydrolyzing peptide bonds in proteins. And so proteins are where a microorganism is going to get these amino acids from in its diet. And so many prokaryotes secrete enzymes that degrade extracellular proteins. And so this is both good and bad in terms of human disease, if we want to take that perspective. But really what they're after is that amino acids are great substrates for food. And so again, by removing these amine groups from amino acids, the remaining carbon skeletons can immediately be used in pathways used for their production. And so I'm taking you first through the process of eating the amino acids because that's going to help us understand how to make them. And so the notable exception here are aromatic amino acids, but that is a subject for biochemistry and I think gets beyond where I want to take you guys in this course and gets away from the sort of core principles that I want you learning here. So, like I said, what needs to happen for amino acids to allow them to be entered right into various pathways we've just talked about in prior lectures is that we need to remove the amine groups from amino acids. And so there are a number of different ways that this is done. So there are amino acid hydrolases that will remove amine groups from very specific amino acids. There are amino acid dehydrogenases that will, instead of using essentially quinones and water to essentially oxidize off this this amine, what can happen instead is that an oxidoreductase enzyme, similar to what we've talked about, is able to oxidize this carbon, liberating ammonia, and in the process creating this this carboxyketo group, which we should recognize some of these from as intermediates from the citric acid cycle, for example. So those two other mechanisms that I showed you are very, very specialized, and there are some specific amino acids that have dedicated deamination mechanisms, but the most common mechanism is a process that is coupled to glutamate dehydrogenase and transaminase. And so transaminase is an enzyme that will take amino acids, and they're very promiscuous in what they will accept as amino acids, and that they will remove that amino group to produce keto acids, like the ones that I just showed you. And what happens is that transaminase uses this intermediate oxoglutarate and turns that into glutamate. And oxoglutarate is essentially alpha-ketoglutarate from the citric acid cycle. Glutamate, then there's a dedicated glutamate dehydrogenase enzyme that recognizes glutamate and performs that oxidation reaction that I just showed you using either NADP or NADPH as the electron acceptor, an electron carrier of that reaction. So the benefit here is that this whole set of reactions relies entirely on glutamate and glutamate dehydrogenase, but essentially has a very, very promiscuous transaminase that allows one process to deal with more or less all 20 amino acids. And that is actually remarkable. So instead of having 20 different enzymes involved in specifically removing amine groups from amino acids, you can essentially have one process to take care of all of this. So where do these amino acids go? So if I were to remove amine groups from their various amino acids substituents, what I would see is that the aromatic amino acids, again beyond the scope of this course, are fed into the pentose phosphate pathway through a series of reactions. Serine, glycine, and cysteine, these all end up as glyceraldehyde 3-phosphate and can be oxidized through stage 2 of glycolysis. Alanine, alanine, valine, and leucine can be fed in to glycolysis as pyruvate, all ending up in the in the citric acid cycle. Now, glutamate, proline, glutamine, arginine, asparagine, lysine, methionine, threonine, isoleucine, all of these, when we remove their amino groups or extra amines and various other side groups, what we end up seeing is that these are all intermediates of the citric acid cycle, can be immediately fed into the citric acid cycle and oxidized. So this is going to tell us a lot of information actually about where these go into carbon catabolism, is actually telling us indirectly how they're made from these carbonaceous intermediates of metabolism. So where we're going to go next is how these things get made.